Dum-ba-dum-ba-dum-ba-dum. Here we go. Hello, hello, hello. Hello. Can hear me? My computer is being a bit slow. Okay, we're going to start in a second. Right, okay. If you are here, that's brilliant. Let's see if this works. Well, yeah, this is my first live stream, so it may not be brilliant, but I'll try my best anyway. And hopefully you're here because you're doing AQA A-level biology paper 1 tomorrow. I'm just going to go through some of the key things, and then I'm going to go through some exam questions as well. Okay, so I'm just going to share something with you. So here we go. So first of all, I want to look at tissue fluid. It hasn't come up for quite some time. So hopefully that you can see that. There we go. Okay, so formation of tissue fluid is quite an important one. Okay, and particularly in terms of 5 markers. So as you can see from the arterial end, you've got in the capillary, you've got the arterial end and you've got the venule end. And what's happening there is basically several different things. So let's split it up first of all. So first of all, the yellow if we look at the yellow arrow, which is hydrostatic pressure, so that's the amount of pressure, is greater in the capillary at the arteriole end. So this means there's an overall outward pressure. So it's against the osmotic potential. The osmotic potential is the blue arrow, and so therefore it's greater. So the hydrostatic pressure forces fluid out of the capillaries and into the spaces around the cells, the interstitial cells forming tissue fluid. Right, I don't think you can see that. There you go. I think you can see it now. Right. Okay, it's working now. Right, so spaces around the cell forming tissue fluid. Now, obviously, the large proteins don't come out, okay? So large proteins are too large. The large, like red blood cells, they can't get through the gaps in the capillary. Okay, and obviously oxygen, nutrients, amino acids, and water get taken out of the arterial end. At the venial end, the hydrostatic pressure now is much reduced in the capillary. The other thing is, due to fluid loss and an increase in concentration of plasma proteins, which stay in the capillary, the water potential is now lower. Okay, and this means that some water re-enters from the venial end, and excess tissue fluid is drained into the lymphatic system. So that's the first thing I want to go through. Second thing I'm going to just mention, which I think is quite popular, is the following. So let me just share this with you. Okay, so here we go. So I think something on monoclonal antibodies. This spec is AQA. It's AQA A-level paper 1. Saying that, there is similarities between all the specs. Hope that helps. So obviously monoclonal antibodies. So we need to understand how monoclonal antibodies could be used. It could be used in things like pregnancy tests, HIV tests, drug testing, cancer treatment, treatment of heart disease. So the key one is the pregnancy test. So if we just go through how a pregnancy test works related to monoclonal antibodies. So at A, you can see HCG, which is a hormone produced in pregnancy. You don't need to know the name of it, but you'll notice basically on the stick, you urinate on the stick and the urine moves up. Well, in this case, down the stick. And you'll see at B that the antigens bind with the antibodies. So you have an antigen antibody complex. Okay, and then it continues moving through to C. Now, C, there is another set of antibodies that are immobilized. In other words, they can't move. So the antibodies will then latch on to that antigen, onto the HCG antigen, and therefore sticking it on. And you can see also on the antibody, what's stuck on the monoclonal antibody is this blue dot. That blue dot could be an enzyme, it could be something that changes color. And in this case, if there is HCG present, it will then give a blue line indicator. It then moves on, and any free antibodies in the pregnancy test will move through, okay? And you'll have a secondary antibody that attaches to the antibody, and that's the control. That's just showing that the actual and the monoclonal antibodies are working. Testing for diseases, HIV. Oh, you can't see again. You're right. Oh, dearie me. Can you see now? No. I need to share. A minute. Can you not see that? Well, you gotta love YouTube. Nope, I can't see it either. Oh, there we go. Hooray! We can see. Thank you. Okay, I think I'm just gonna well, I think I've explained the pregnancy test, not very well, but we'll go on. So testing for things like diseases like HIV. Okay, you can't see that. It's terrible, isn't it? Can you see that now? Yes, you can. Right, I'm going to stay there. All right, so you need to mention something called the ELISA test. The exam board, AQA, it's AQA. So the ELISA test, they might ask you about. So this is using lots of monoclonal antibodies to test lots of different samples to see if someone has a disease. So what they do is they bind the antigen that they're interested in. The HIV antigen is bound to the bottom of the reaction vessel. So the reaction vessel is usually a well, a plastic well, and there's lots of different little indentations in it. And then the antibody, not specific to HIV antigen, and you've got an antibody specific to the HIV antigen, which is the primary antibody. So you add the blood sample, okay? And if HIV is present, it will attach to the antibody. Primary antibodies will attach to the antigen. You have to wash out, you have to wash the excess out, and then a secondary antibody with the attached enzyme attaches to the initial antibody. You wash it out, and then there's a color change, and if it's present, you get a positive result. So in an exam question, they might actually ask you, why is this quantitative? Well, color change is not usually quantitative, but what you could do is you could have a range of concentrations or of, for example, HIV, so you know what colors there are, and you could use a colorimeter, and that colorimeter would tell you the absorbance, and you could use a calibration curve, and that's something that they to do. Right, okay. Another thing I wanted to show you very, very quickly. I don't know whether it's going to work. Right. I'm going to go through some questions, actually. I think that will be the best thing to do since you've got your exams tomorrow. So this is last minute stuff. I've chosen these questions because I think that they might come up. So I'm just going to find those questions for you. Let's see if I can find them. There you go. Here you go. So hopefully you'll be able to see these. There we go. Yeah, I can see them. Good. Right. Okay. So just very, very quickly then, cohesion tension, definitely cohesion tension theory will come up. Okay. I think cohesion tension and mass flow will probably both come up. So describe how water is moved through a plant according to cohesion tension theory. I hope this is going to work. Let's see. So how is water moved through a plant? Well, basically water is, first thing to say is water evaporates out of the stomata. So that is transpiration. So water evaporates at the stomata. Okay. It's the first thing. Hopefully you can see that. Can we see that? Yeah, it's writing it. That's good. Okay, so water evaporates out. Basically, that reduces the water potential in the cells. Okay, in the cells. And water is drawn out of the xylem because it's moving from a high to a lower water potential. That creates tension. And also you need to know about principles of water properties. So there is cohesion forces between the water molecules. So there's probably more than you need there. Okay, to be pulled up the column. So those water molecules pulled up the column. So I'll just give you a moment just to write that down. Excuse my writing. I'm going to get a better tablet. Okay, you got that? Hope that's okay. So that's the first thing I wanted to show you. So cohesion tension, I think, would be good. Right. The other thing that I think as well might come up is about hemoglobin. Okay, so loading of oxygen. All right, so I'm just going to quickly show you, hopefully show you PowerPoint. I'm going to have to get better at this. I know I am. So I'm just going to share this video. Okay. Right. Okay. So I just want to mention about the partial. Oh, it's gone black again. Look at that. It's terrible. There you go. You can see that. So I just want to mention about the partial pressure of oxygen. So underneath me there on the X part of the graph, partial pressure, and then the percentage saturation of hemoglobin with oxygen. Okay. So HB has a higher affinity for oxygen in oxygen-rich areas. Okay, so in terms of, for example, the lungs. All right, HB has a lower affinity for oxygen in oxygen-starved areas, such as things like respiring tissue. So there's a lower partial pressure promoting oxygen unloading. So this ensures oxygen is always picked up by red blood cells in the lungs and released in respiring cells. So if, for example, a animal is really highly adapted to living in high altitudes with very little oxygen, so you might find that the curve would be more to the left, for example, okay, because at lower partial pressures they pick up more oxygen. There's a higher oxygen saturation, okay, and also it's basically it can pick it up faster as well. But I want to talk about hemoglobin. So describe the role of hemoglobin in the loading, transport, and unloading of oxygen. Right, it's gone black again. Right, I think it's better when I go to the Word document. So here you go, the Word document. So this question here. So this is a 5 marker. So this is the end of paper 1. Hopefully you can see that. No, you can't. Yes, you can now. Okay. Right. I'm just going to go through this 5 marker with you. All right. So as we've already said, we know that HB, okay, it loads or associates with oxygen. Okay. So wait a minute. Okay, so it loads with, you seeing that? Okay, so HB loads or binds with oxygen. Yes, you are seeing that. That's good. And it binds at higher partial pressures, so higher partial pressure of oxygen. Now, the thing about hemoglobin is, okay, that basically you get that, so it binds, the oxygen binds, so the first kind of molecule is basically is quite hard, but as basically it increases because it gets easier, so it gets easier for oxygen to bind with the hemoglobin, okay, and it forms oxyhemoglobin. Now, then it can unload or dissociate in the cell. So it can unload, okay, and it unloads at a low partial pressure of oxygen. Now, I have missed out one thing. It says in the transport, well, oxyhemoglobin, it's going to be transported in the red blood cells. Okay, simple as that. Sarah, yes, you do have to say that water is drawn upwards. Okay, so upwards from the xylem, and the water column is joined together due to the cohesive nature of the molecules of water. That's great. If anybody wants to ask questions, please do put them in the chat. That would be great. Thank you. Okay, another quick one is protein synthesis. So transcribing, translation. So I've got a nice question about translation here. So I'm going to show you that in a second. Lena, yes, you can, but in AQA we wouldn't necessarily use those words. Well, I wouldn't. But if your teacher's told you to use them, you use them. Okay, that's something I'm going to point out. I am a teacher, but I know you've all got your own teachers. If your teacher's taught you in a specific way to say specific things, you do that. Don't do what I'm telling you. Okay, I'm just going through generally what would be in the mark scheme. All right. You don't need to learn the formula for standard deviation. Okay, so standard deviation, what you need to know is about, you might be shown the bars for standard deviation. Okay, and then you need to know if they don't overlap between two things. Okay, let me show you. I want to show you because I actually tutored a student this evening and we discussed that. So wait a minute, Tom. Let's see if we can help you here. Right. Here I've got, right, two bars. It doesn't matter what the bars are. And I've done standard deviation plus and minus 2 standard deviation. Okay. If the bars do not overlap, that means there is a significant difference between the two samples. Okay. And you would say, for example, if they asked you to evaluate a particular experiment, you could say, well, in this experiment, there is a significant difference as the two bars do not overlap. If they did overlap, so if I redo them, if they do overlap, so they're equal both sides, like that. If they did overlap, then that would be not significant, and therefore there is no significant difference between the two samples, if you get what I'm saying. But you would never be asked to do formula for standard deviation. Formulas where you would need to know about index of biodiversity, even calculations related to microscopy. They tell you the area of a circle, which is pi r squared, stuff like that. So they'll give you most of the stuff, to be honest. Good for you, Ruben. Right, okay, I'm going to go through translation next, okay? I think that's good, actually. What I'll do is I'll go through something and then you can ask me questions. That's a great idea. Right, describe. Oh, let me just check you can see it. Here you go. Describe a polypeptide. How a polypeptide is produced by translation of mRNA. Okay. So let me just get the right thing. So what we're looking at related to this thing is translation, and it's a 5-mark question. And you would have to, first of all, you would not talk about how mRNA is produced, because that's transcription, transcribing, whereas this is translation. So the mRNA basically would travel to the ribosome. Okay, now I'm just, because my writing is really rubbish, I'm going to just write the main thing. So RNA travels to the ribosomes, okay? All right. Or you could say the RER, that's fine as well. They'll accept both. All right. And then what you have, you've got tRNA and the anticodons are complementary to the mRNA codons. Okay, so anticodons, complementary, that is a key word you're going to use all of the time. There's lots of words that use loads of things that use complementary. So anticodons, they are complementary to codons on the mRNA. And you need to mention the tRNA brings the specific amino acid. Okay, so the specific AA amino acids. And then what happens is the AAs, they join by peptide bonds. You might want to mention as well, you can mention the triplet code as well if you wanted. It's a triplet code for each amino acid, but joined by peptide bonds. How are they joined? With ATP? Okay, and then tRNA is released and the mRNA moves on. Sorry, not the mRNA, the ribosome moves, not the mRNA. So the ribosome moves along and it reads the mRNA and it forms a polypeptide. Simple as. There you go. Okay. Forms polypeptide. Hopefully you can read my terrible writing. Right. Okay. How would I answer it? I like you, Ruben. You asked me some good questions. So here we go. So about transcription, I'm going to do it on here. I was going to have something over my head, but it didn't actually work. So nothing much is working very well on this, to be honest, but at least you can see that. Right. Okay. So transcription. 5 marker. Right. Okay. So transcription, I might just write it actually. Yes, this is for AQA. This is for AQA. It's for AQA. For tomorrow's AQA. Right, I'm going to write it here. So, right, so transcription. Right, so first of all, okay, the DNA, okay, the DNA hydrogen bonds have got to be broken. So hydrogen bonds broken. I think, and you'll probably put me right if I'm wrong, it's DNA helicase. Okay, so DNA helicase breaks those bonds. That's number one. Secondly, okay, the double strand separates. And it exposes the bases. Okay, and then free nucleotides in the nucleolus nucleotides. They combine with the complementary pair. Is this for A? Yeah, it is. It's for AQA. I'm just having a quick drink. I'm a bit thirsty. Okay, so one of the, you could say a combined, well, you could say one of the strands is a template strand, basically. So you get 3 nucleotides, get specific complementary base pairing. So I didn't say base pairing, did I? Complementary base pairing. Okay. So, and you do need to know that the base T is replaced with uracil. Base T is replaced with U, which is uracil. Can say U, that's fine. Okay. When they paired up, they then join together by RNA polymerase. So RNA polymerase joins them up. Okay. And then it assembles the mRNA strand, so it separates and it assembles the mRNA. Now, mRNA is very short, okay? It's usually 1 gene, but it's usually quite short. And then the DNA, the strands of the DNA rejoin. So strands of DNA rejoin. This is more than a 5 marker. Reform into a double helix. So the mRNA, it contains introns and exons. Okay, so it's known as pre-mRNA actually. And then what happens is they are spliced, and then once they're spliced, they can then move through the nuclear pore. Okay, and then they move through into the cytoplasm. Pretty much that's it. Okay, right. I like this one as well. So cell fractionation and ultracentrifugation. Before I do that, I'll come back to you guys. Yeah, it trips me out a bit, to be honest. Right, so good. Right, have we still got people here? This is going on for tonight. You need to get a lot of sleep as well. That's important. Right, good. Okay, here we go. So cell fractionation and ultracentrifugation. There might be a shorter question, might be a longer question. You never know. So if you noticed, I'm going through quite a lot of long questions, but this is quite good for your revision to revise large topics, if you see what I mean. So let's just go through ultracentrifugation. Well, describe and explain. So the command words there are describe. So you got to say what happens, but you got to say why it happens. So why that it happens. And they want you to talk about both cell fractionation and ultracentrifugation. So first of all, how are you going to get into the cells? Well, the key word is homogenize. I do like that word. It'd be homogen. It'd be good if I could spell it. There you go. Homogenize, and it releases the organelles. Okay. So you can they just use a blender basically to break them open. Don't forget this stage. You've got to filter it then to remove the cells or tissue debris. Okay. Then in the solution that you've got, okay, it's going to be cold. Well, why does it need to be cold? So that's a description. It's a cold solution. Why? Because it prevents enzyme activity. So it prevents enzyme activity. It's also what we call isotonic. So isotonic just means the water potential is equal. So an equal WP. Why is that? Well, it prevents osmosis because you don't want your samples to shrivel or expand. So it prevents osmosis. Then you're going to talk about the, well, basically it's cold, isotonic, and you'd also put it in a buffer as well. So buffer the pH. Why? Because that would stop enzymes from denaturing. Okay, so that's quite good for the fractionation aspect. Then we've got to talk about ultracentrifugation. So you centrifuge basically, and if they don't ask you a big one, but they might ask you this about centrifuge. So you centrifuge at a low speed, first of all. Okay, so a low speed. And what happens is that the nuclei in the pellet, so that's the most dense, all right, will go to the bottom of the tube. So it moves to bottom of the tube. Okay. So that's separated out the nucleus. Okay. And then what you do is you remove the supernatant. So that's the solution above the nuclei. So basically you would have at the bottom, you've got a pellet, that's the nucleus. And then you've got this, which is the supernatant. So remove the supernatant. It's the name of my rock band. Well, it will be. Supernatant. That sounds good. And then you would centrifuge at a higher speed, and then you can separate the next organelle. So it's the higher the speed, the less dense it is, basically. Less dense organelle. So you go I think it's nucleus and then other large things, depending if it's plant cells, but it's chloroplasts, mitochondria, and then you're getting into kind of smaller stuff like RERs, might be Golgi, but then getting smaller and smaller to things like vesicles. Okay, right. Good. I hope you can see that. All right, so that's cell fractionation, ultracentrifugation. Okay. I'm going to go through a bit about proteins next. Yeah, I could do, but I'm not going to tonight, I'm afraid, Lena. Sorry. I think, mate, I'm going to do something similar for paper 3, and when I do paper 3, it'll all be graphs and tables of data. Okay. There will be some graphs, obviously, in paper 1, but not loads. I'm just trying to get through a large amount of content at the moment. Hope that answers your question. Right. Ooh. Okay. So I want to talk a bit about proteins. So we're going to go through proteins now. It's going a bit better, isn't it, pals? It didn't start off very well, but it's improved. Good. I'm glad it's beneficial. That's what I'm trying to do, is to help you. So, right. So, okay. Let's look at proteins. So here you go. This one here. Describe how a quaternary protein is formed from its monomers. Do not include the process of translation. Well, we've already talked about translation, but I'm not going to talk about that. Now, proteins, they come up every single year. Okay, there will be proteins in there somewhere or another. Okay, so quaternary proteins, well, you've got to say, let's start off with the basics. Well, the basics are amino acids join by peptide bonds. So you've got to say that peptide. Trying to improve my writing. Peptide bonds. Okay, how do they join though? You could say by condensation. Okay, so a condensation reaction. All right, so where a molecule of H2O is released. Then how do you get the secondary structure? Okay, so you'll want to know, well, how do you get the secondary structure? Well, for the secondary structure, it's hydrogen bonds. So hydrogen bonds form. So you get hydrogen bonds forming the secondary structure. So it's asking for all the things, okay, going through the different structures. And then tertiary structure is formed, okay? Tertiary structure with the interaction between the R groups. So interactions between R groups of the AA. Okay, so that's only one polypeptide at the moment, but we know quaternary structure is formed by the interactions between polypeptides. And you could actually talk about disulfide bridges, ionic bonds, that kind of thing, okay? So it contains more than one polypeptide. So quaternary is more than one polypeptide, greater than one polypeptide. Joined together. Actually, that reminds me, I'm going to talk about antibodies in a minute as a good example as a protein. So you've got them held by H bonds, ionic bonds, disulfide bridges. Disulfide bridges. That's all it'd be looking for in that. Bridges. Right. I know this is not about antibodies, but I'm just going to I want to quickly talk about the structure of antibodies. Okay. So an antibody is obviously a polypeptide. It's made of a chain of more than one polypeptide. Okay. And you've got a constant region that doesn't change. And you've got a variable region that does. Okay. So you've got your variable region there. And basically, you've got receptors sites that bind to a complementary shaped antigen. So here is a bacteria, let's say, and it binds on to it's complementary to the shape of the antigen. Okay, so you can form antigen antibody complexes. Okay, and the shape of the antibody and the shape of the receptor sites is controlled by the amino acid sequence. So amino acid sequence is important for the shape and the structure. And as we said, they're held together by peptide bonds, hydrogen bonds, disulfide bridges. You can mention lots and lots of different structures there. Right. Bit of a mess, but there you go. Right. Okay. Right. Moving on quickly because time is against us. I haven't talked about genetic variation. I haven't talked much about mutations. So and also gas exchange. So one of the things that has not come up for many, well, not many years, but a few years is insects. And I'm going to talk about insect gas exchange. Before I do, though, I want to talk about mutations in genetic variation. Okay, so this is very much about how meiosis occurs. Okay, so how do you get genetic variation within a species? And it's going to be things like meiosis, random fertilization, new combinations of alleles. All right, so I think as you're probably all aware, Miss Estruck has been talking about mitosis and meiosis quite a lot. She's my pal, so that's good. I hope we're friends anyway. So let's go through that now. Right, okay. So here we go. Describe and explain. Right, okay. Mutation results in an increase in genetic variation. Describe and explain the other processes that result. So actually, in the previous question, it was talking about other processes, but it was talking about another process, but I'm not going to talk about that. Okay, I'm going to talk about this one. So the first thing is you get independent segregation of homologous chromosomes. Segregation of homologous chromosomes. Homologous meaning a pair of chromosomes. There we go. You then get, okay, so if you get independent segregation, that gives genetic variation. Okay, you get crossing over as well. So crossing over between homologous chromosomes. So that's on the chiasmata, I think. And you've got to mention between. Notice the detail you've got to give. You can't just say crossing over. You've got to say crossing over between the homologous chromosomes. Okay. So I would say that's one top tip from me is write detail. Just write the obvious, write everything. Okay. Don't miss things out because you think, well, the examiner knows what I mean. Just write. And if you don't think you need to write it, write it. Okay. We also know that gametes, sperm and the egg, so there's random fertilization of gametes. That creates variation, definitely. Random selection. Okay, and therefore, what will that do? That then produces new combinations of alleles. Okay. And I'll put it in big letters, alleles. Whenever you're talking about genes and genetics, I'd always talk about alleles. Okay. You don't want to kind of, it is alleles, always alleles. Okay. There we go. All right. If there's no questions after that, I'll give you a moment longer. TMR. What's TMR again? I should know. So genetic variation, okay, does come out in paper 2 as well. Right, I do want to talk about spiracles and insects. Yeah, it is tomorrow. That's why I'm going through it now. Okay, I've only got 10 minutes as well, because as I said, you lot are going to need a bit of a relax before you go to bed. Right, okay. Well, you can do what you like. I'm not telling you what to do. Right, okay. So this one, question 5. All right. As I said to you, gas exchange, insects has not come up for some time. All right. So I am going to go through this because many students forget about insects. I would know a bit about leaf structure as well, which hopefully you do. Just check that you can see that. Just don't trust YouTube. There we go. Right. Okay. So describe how the structure of an insect gas exchange provides cells with sufficient oxygen, but also limits water loss. Explain your answers. Okay, so you've got to talk about the structures first. All right, so you've got things like you could mention the spiracles. So these are the holes on the outside. You can mention the tracheoles, or okay, I would say trachea, or I'd say tracheoles. What do the spiracles do? Well, they allow diffusion to occur. Okay, so they allow diffusion of oxygen through the tracheoles. Okay. The thing about the tracheoles is that they have a large surface area. Okay, so they're highly branched. Why is that useful? Well, that means that they have a large surface area. Okay, for exchange, large SA for exchange. Okay, now obviously with this subject, with anything related to gas exchange, there are always three things they want you to talk about, regardless of whether it's fish gills, lungs, or whatever. So you've got to talk about large surface area. You've got to talk about why it's thin. Well, the tracheoles are thin. All right, why are they thin? Okay, explain it, because there's a short diffusion pathway. Okay, short diffusion pathway, or distance, actually. Pathway or distance. Short diffusion distance, actually. Yeah, that's better. Let's move that. Yeah, short diffusion distance. Okay, the tracheoles are also permeable to oxygen. So they're permeable. Now, in terms of limiting water loss, the spiracles can close, okay? So they can close to reduce water loss, okay? Or they have valves to prevent water loss. You might not know the following, but they've also got hairs around them. So if I draw the abdomen of a bee or a wasp, I don't want to worry you or anything, but the spiracles, they also have hairs coming out of them. Okay, now hairs are quite useful, okay, around the spiracles, because that reduces water loss. So hairs reduce water loss. There you go. So there's lots there, but you get the general idea. And it's the same for things like, I don't know, fish gills. If the fish gills do come up, countercurrent multiplier. Why is that good? Well, it maintains a steep concentration gradient, for example, and therefore all of the oxygen from the water is being absorbed into the fish gills, for example. Okay. Right, there is one last thing I do want to go through in a minute, and that is the properties of water. Okay, can you say tracheoles are 1 cell thick? Yes, you could. That's a good idea, Lena. I like that. K, yes, they have AQA A-level paper 1 tomorrow afternoon. Lucky them. I like paper 1. It's good. It's easy for me to say. Right, so yeah, one of the last things I want to go through. Yes, you could say that. Oh, very good, Lena. Yeah, they do have a thick waterproof exoskeleton. Yeah, I don't know, Pels. I haven't heard about that. I wouldn't stray into things that are out of spec. AQA really like you to use the specification, so I don't think fluid filler ends is in the spec. What practicals am I predicting? Do you know, I'll be honest with you. I think at this stage, you just got to know pretty much everything. Most of the practicals you've done anyway, but usually they ask you in a different way, and they usually give you lots of information about the practicals anyway. It's usually looking at control variables, independent variables, dependent variables, things like that. Okay, why is it a good experiment? Evaluate it. stuff like that. Well, that's great, little cranky. I don't think I'm going to be helping you with media. You know more about media studies than I do, mate. Okay, so I don't think you need to be here. Unless you're just interested, that's completely fine with me. Right, okay, so last one I want to go through. Here we go. Water. Okay, now Miss Estruck has been going on, hammering on about this recently. Okay, so about the properties of water. It does come up. Whether it will come up as a big thing, I don't know. But they do like you. They haven't mentioned a lot about properties of water. Also, semi-conservative replication. If we've got time, actually, I want to talk about semi-conservative replication, and then I think we'll call it a night. But here we go. AQA. This is AQA, Grace. AQA. Yeah, it could do. I thought chylomicrons, I think you mean. That's a typo. So about lipid absorption, yeah. Chylomicrons, bile salts, yeah. I think something on viruses will come up as well. So virus replication, structure of virus. Right, okay. 5 properties. Let's just go through this, all right? Because you're probably all going, get on with it, sir. Get on with it. Right. First of all, then, okay, it's a solvent. So properties of water, the first key thing is, I think, it's a solvent, okay? So, because I would always use that word, solvent, so reactions can occur, okay? That's all you need to say. That's 1 mark. You will only get 1 mark for that. You don't get 2 marks. You've got to say, you've got to give the property and what it does for 1 mark. 2nd mark, okay, there's more than 5 marks here you could get. It has a high specific heat capacity or high heat capacity. Why is that important for organisms? Well, it buffers the changes of temperature, so you don't get extremes. Okay, so it resists the changes in temperature. You get stable, homeostatic internal environment. But I'm veering onto paper 2, so we won't do that at the moment. I'll do this. I will talk about homeostasis after you've done paper 1. Large latent heat of vaporization. Why is that important? Well, in organisms, that has a cooling effect. Okay, so you get evaporation of water, okay, and you get cooling effect, which is useful adaptation. Number 4, we've already talked about it, would relate to water cohesive nature, so cohesion. Why between, you don't even have to talk about it being, you could mention about it. You have to be careful not to go into too much detail. If you're a chemist, you don't need to go into loads. Okay, between the water molecules. All right, what does that do? It just supports columns of water in plants. Okay, could mention even they'll accept cohesion tension theory as well, and also cohesion again. Why is that useful? You get surface tension. So surface tension is useful for supporting small organisms, supporting small aquatic organisms like pond skaters. Okay. You know, there's you could have lots there. What else could you have? No, that'll do. That'll do. Okay. So properties of water. Yes, Kimberly, you got to stick to paper 1. Definitely. It's all about the spec. I tell my students that it's what's in the spec. So it wouldn't be marked wrong. Can I also say you never marked wrong? It's just not marked. You don't get a mark for it. Yeah, they never take marks off you. Good point, Lena. You're a good bunch of students. I'll be very impressed with you tonight. Right. So to answer Lena, continuous column. I don't think you have to say a continuous column. No, you don't. Just supports column of water. Is there a difference between solvent and metabolite? Generally, yeah. So a metabolite in a condensation reaction, so condensation hydrolysis. So yeah, it's different. You're right. Well done there, Vanessa. So you could have mentioned it's a metabolite in condensation. So it allows reactions to occur, photosynthesis, respiration, stuff like that. Very good. I think you're all going to do very well tomorrow. Okay. Do you want me to go through semi-conservative? If you do, put your thumbs up. If you've had enough, we put your thumbs down. Will this work? Right. Yes, okay. Since you were here first, pals, we're going to do semi-conservative replication. Good. Okay. Cool. All right. So, right, semi-conservative. This is related to, obviously knowing what semi-conservative replication, but the kind of the evidence behind it. So that's the idea of Messelson and Stahl. And I will talk, even though this is not about Messelson and Stahl, they do like you to know about semi-conservative replication and looking at test tubes of bacteria that have been grown in different types of different types of nitrogen, whether it's heavy nitrogen or light nitrogen. So we'll talk about that. Right. Anyway, very quickly, because I've got marking to do tonight. I'm marking GCSEs. So there we go. Right. Yeah, I'm doing one more question, Lena. That's great. Thank you. Right. Describe the process of semi-conservative replication. Right. Okay. So this is the ideas of, it was Watson and Crick who came up with the idea. Messelson and Stahl actually did the experiments. Okay. Right. DNA. Oh, wait a minute. Oh, I'm having a mare. There we go. No, we're all right. We're okay. Right, we're okay, guys. Right. So the first thing is good old DNA helicase comes along. So DNA helicase splits those double helix. So it unwinds or breaks the hydrogen bonds. So it unwinds the DNA helix. Getting much better at my writing now. I should have practiced earlier. Okay. Or it breaks hydrogen bonds. That's what I probably would have said. Both strands act as a template. So obviously when mRNA is being produced, you've only got one template strand, but in DNA replication, both strands are a template. Okay. So you get exposed bases basically, and then 3 nucleotides line up in complementary pairs. So you have complementary pairs. You all must know this. The way I remember the complementary pairs, apples are found on trees, and usually cars are found in garages. Well, they're not always, but you know what I mean. A to T, C to G. Okay. What joins them together? Well, DNA polymerase. Okay. It's not RNA polymerase, it's DNA polymerase. And this joins the nucleotides. Okay, and what do? They form phosphodiester bonds. So getting all those keywords in, guys. They love all these keywords. Use them. Okay. And then the key thing is each new DNA molecule consists of an original strand. Okay, so it consists of one original strand. Okay. I just want to mention Metelson and Stahl. So they did the experiment. Okay. Let me just find a space here. Wait a minute. Right. M&S. You don't need to know about Metelson and Stahl and their names, but I like their names. So what they did is they took Petri dish and they grew some bacteria in the Petri dish, and it had in 15N, which is heavy nitrogen. So it's a heavy isotope. Okay. All right. And then they centrifuged it and they found the 15N to in the, ooh, what the hell's happened there? Okay. They found a strand like that. Okay. They then took the 15N bacteria, okay, and they put it into agar that had 14N. Okay. All right. So they took the bacteria from there that they produced and put it in the 14N, and then they allowed them to replicate. Oh, God, that's awful, Dr. Burgess. Dr. Biology, sorry. Right, let's do that again. Oh, God. Can't draw. Right, I'm trying to draw a test tube. There you go. Right. So what happens is, it's a shame I haven't got different colors. So the original strand, here's the original strand of DNA. Okay. It splits and it only has one of the original strands in each. Okay. And then, can I change the color? Let's change the color. Hooray. Will it work? You get the new strand. So this is the new strand that contains the 14N. There you go. Okay. So what happens is that then if you were to centrifuge it, oh, wait a minute. Is that coming up? Yeah, it is. Right. So you centrifuge it, you would get one smaller line there. Okay. Because, well, no, not smaller line, same line there, the same distance, because it's still got two strands of the original DNA, but it's also got two strands of the lighter DNA strands. So therefore, it will have another band, and that band will be higher up because it's less dense. Okay, so I'm going to draw yet another test tube, because then the next bit that they did, okay, they did this. So let's split it up again. So this is semi-conservative replication. So I will have my original strand. Okay, so I've got one original strand there, one original strand there, and then just change my colors because I've got to have the new strand of the 14N. You would have it there, okay, and there, because they've split up. But if you then go on to culture them, okay, and continue in 14N, you've got a new strand there, new strand there, terrible double helix, but there you go, can't do everything, and there. So now we have, if we were to then centrifuge them out, we would have the original strand there, okay, but we have a lot more new strands, okay, so they're less dense, so they'd still be higher up, but this band would be really thicker. It's thicker because it's got more 14N in it, like that. So that's kind of a popular exam question there related to Metelson and Stahl, and they get you to look, say what happens to the test tubes. Okay. Some of this is applicable to EdXL, yes. Okay. Right. Okay. So any more questions? Oh, I wanted to show you my t-shirt. Look. I might look like I'm doing nothing, but at a cellular level, I'm really busy. Do you like my t-shirt? It's cool. Anyway. Good point, Lena. Right. Evaluate. I love evaluate. Evaluate. Pros and cons, for or against. Okay, so evaluate is always looking at both sides, usually related to an experiment. So practical, for example, they might give you lots of information, and you need to look at things like, you know, is there enough people or enough things that they're actually sampling? Have they repeated the experiment? Have they controlled the variables? If they're using humans, do we know the gender? Do we know their mass? Do we know their medical backgrounds? Stuff like that. Okay. All right. So evaluate always comes up. Paper 3 will be full of evaluate stuff. Okay. This is AQA, Veniac. Okay. Right. Okay. Well, I hope you found that. I'm going to stop sharing. Hope you did find that useful, guys. Remember, just a few exam tips here, okay? Read the question very carefully. What are the command words? Have you looked at all the information? There is time. You do have time in the exam to look at the stuff that you're being asked to do. Some students, they like to do the 5 markers first. So if you look at the back of the paper, you go, oh, I know that. I'm you know, something comes up tonight. By a miracle, it'd be great if it did. Then you go, right, I'll do that straight away. Okay. You don't have to do the exam from start to finish. You can decide how you're going to do it. I'm pretty sure, you know, all of you, if you're doing A levels, all of you know how to do exams now. And you have got this in the bag. You might not think you know everything, but you probably don't know everything, but you know most things, and you've got this far. So I'm pretty sure, you know, you'll be fine tomorrow. If you're watching stuff like this and you're interested already, you should be okay. All right, that's great. Definitely, Kimberly, correlation does not mean causation. There was a good thing in the news about that, about eczema and having dogs when you're young and you don't get eczema. That's a correlation. Okay. All right, guys. Well, I'm going to say goodbye now. Okay. So I hope you found that useful. And I wish you all the best. I'm going to do this for paper 2 as well. So I'll let you know. I will set that up a bit earlier than I've done tonight. It'll be the night before again. I might even do something before that because this has gone very well. All right. So thank you, guys. Okay. I'll see you soon. Take care. Keep going. You got this. Bye.